So to finish off this morning's session, in my mind, I kind of see these two talks as sort of moving into the future and sort of moving beyond the ways we've been thinking about these technologies. And to start that off, we have Dr. Lynn Rothschild, who's come from NASA Ames. She's a synthetic biologist there and an astrobiologist, which I think is a pretty groovy term. And she is going to be talking to us about synthetic biology on Mars, designing solutions for Earth.
Thank you very much. I was tempted by the end of the last talk to completely throw away all these slides and do something different, because I do feel a little bit as maybe the one fed in the room that I've been a little beaten up on behalf of the federal government today. And is this being recorded?
Oh, yeah.
All right, well, I'll make the we're not this monolithic evil organization that some of you are painting us. It's a little bit more McHale's Navy sometimes, and I'll be happy to talk about it more in the panel. But it's not quite what it was portrayed. And I'll just say that there was a regulatory meeting of just representatives of different federal agencies at NIST a couple of years ago that I was fortunate enough to go to. And that's when it became very obvious that there's some agencies that treat one set of organisms as something that is counterintuitive, and another treats them as something else, because many of these regulations have been in place for a very long time, and they don't take into account things like this. And so the point of the meeting that I went to was to try to poke holes in the federal regulations to see where the gaps were. And so there is a lot of work going on right now trying to fill the gaps and coordinate and so on. But again, we are your friendly feds. We're not really the enemy here. Thank you. Any questions? So that's my, advertisement here. So I actually had gone on the assumption that this was going to be a more heavily synthetic biology in terms of the technology. So what I'm going to do is sort of take you back before the first talk yesterday for those people in the room who aren't aware of some of these things that we're talking about, and start with, what is synthetic biology? And the way I like to think about it is that, for example, for hundreds of years we did descriptive biology, well, literally thousands of years, and now we're doing synthetic biology, sort of analogy with doing descriptive chemistry versus synthetic chemistry. Now, there are all sorts of definitions that are floating around, including talking about interchangeable parts and standard genetic elements and engineering principles and so on. But I think that this basic stripped-down definition that we more or less have borrowed from the British is as good as any. It's just the design and construction of new biological functions and systems not found in nature. And what we're talking about primarily are things like novel cell components, metabolic capabilities, certainly the gene drives that people keep alluding to, CRISPR technology, so editing genes and so on, or potentially making a whole new cell. That's the sort of stuff that we're involved in a little bit, is very much in the synthetic biology realm. A lot of what has been known as biological engineering was really synthetic biology. There's no point in arguing how many angels can dance on the head of the pin sort of thing. Now, most people work on applications for the Earth. Manufacturing, environmental control has been a big one at this meeting, pharmaceuticals, fuels, materials, food, and so on. And what I was asked to do when I was told to start this program is look at where synthetic biology could be useful for human settlement, primarily on Mars. We always have to see with the new administration whether Mars is out and the moon is in, or the moon is out and Mars is in. So we try to stay a little agnostic, although these are very, very different destinations. And this is probably as good a definition of synthetic biology as anything. Now, instead of giving an acknowledgement slide at the end, I'm giving an acknowledgement slide up front. Since 2011, I've been the faculty advisor of a combined Stanford and Brown University team of undergraduates called an IGEM team. And what this is, if you're not familiar with it, is a program that started about 13 years ago as a class at MIT to get groups of undergraduates, although it's sort of leached into both the high school now and the graduate divisions, to do a project in synthetic biology biology. There's a huge competition, friendly competition, jamboree, that's then held in Boston every year. This one is fairly late. It's going to be the middle of November. And what the students have to do is come up with a project, do it, including making new bricks. They call them bio bricks, where they're genetic elements that they have to put standardized parts on, so that in principle they're interchangeable, and it makes the whole field of genetic engineering much simpler. But what's interesting, I think, for all of you is that there is very definitely a human practices part of this, and they're judged on it. And I have to say that one of my former IGEMers, Evie, is in the room, who's now a graduate student here, so she can tell you privately how awful it is to work in my lab. Anyway, this competition being something that's fairly open in terms of projects, but being hosting the team in my lab, we obviously have to do things of interest to NASA. So we started off looking at using synthetic biology to make a human-enabled Mars colony. That was in 2011, and then we focused on questions of astrobiology in 2012. And you'll see various projects from these teams, and that's why I'm just telling you up front to put it in framework. That was Evie's team, which was loosely around the idea of biocommunication. I'll mention one of those projects. Then we made a biodegradable drone, which got a lot of press, and again, I'll mention some of the aspects of that project. Bioorigami, which I'm not going to talk about, but it was a very successful project. Then we made a bioballoon last year, which I'm only going to mention in passing. So we've done a lot of very different things where you have a proof of concept using synthetic biology for space applications. So why should you want to do synthetic biology on Mars? Don't we have a big enough planet here? Well, the number one answer is it's my job. I work for the government, and I was told to do that, so I did it. But besides the obvious thing, the reason I was told to do this is because it is what I believe the only ultimate approach for human settlement. You can go like the Apollo astronauts did for a week, and I believe I'm already on record saying you can pack a few peanut butter sandwiches and come home, and that's fine. But if you want to spend a longer period of time, you're going to have to learn to live off the land, which means something very different off planet than it did to the pioneers, or even the settlers in an earlier generation, the so-called Native Americans. When they came here, they lived off the land. They didn't bring everything they needed forever with them. And we're not going to be able to do that when we move off planet as well. But I want to throw in this third point because I think it's more relevant to this group, and that is it also provides novel constraints and freedom from Earth-centric concerns. And that includes the sort of environmental concerns that have been brought up over and over, as well as political ones which really have not been touched on. And what I'm thinking there is, for example, I went to a meeting a few years ago where there were questions about growing cotton and the amount of water that it used. And we weren't really talking about transgenic cotton. I said, well, you could always make a cotton plant that doesn't need as much water, or could use more saline water, so you can mix the fresh water with the ocean water. And I was immediately pulled aside by someone from the State Department and said, no, you don't want to do that. You don't understand that this is soft diplomacy. You want to have the Indonesian cotton farmers fully employed so that they're not susceptible to having, ISIS or whoever coming in. There are all sorts of political factors that are tied in when you make a decision. And then, of course, the economic factors. We are freed, and I'll make this point again later, from terrestrial concerns. I don't have to worry about an indigenous petrochemical industry because there isn't one on Mars. And so that gives me the freedom to try things that you might not have, thinking about Darwin's wedging metaphor, you would not have the freedom to try here. So let me set the scientific background a little bit. This is an actual photograph from the surface of Mars from one of our robotic surrogates. And I'm asking all of you if you can find Earth in this picture. Nope. Okay, there's that little guy there. That's planet Earth. And I love showing this picture because it brings home the point that Earth is going to be this little tiny dot in the sky when we move to Mars, or we at least have long sojourns there. And so you won't be able to be in constant communication. It's not like Amazon.com is going to do next day delivery. This is going to be something remote. Will people think of this as this little data processing center in the sky? Will it be be something that we read about in textbooks, or maybe if you have a particularly, elegant, extravagant honeymoon, you go to planet Earth for 3 weeks. I don't know, but the fact is that the link is not going to be as tight as we sort of are imagining it right now. So there are going to be all sorts of needs for human settlement. You need transportation, habitats, of course, life support, food, oxygen, medicine, and so on. Power, heat, light, and radiation protection is a big one. But our challenges, again, are not the economic ones or the political ones that you would have on the Earth, but are things like up mass and up volume, which actually are very closely tied in with cost. So it costs about $10,000 more or less to launch a can of Coke into low Earth orbit. And at that point, what do you have? A can of Coke in low Earth orbit. That's not going to get you a whole settlement on Mars. It's going to cost you a little extra to get it there. And so any way that you can reduce the up mass, the mass that you're trying to launch against Earth's gravity, is hugely important. And this is where biology can be hugely important as well. Because biology, unlike robotics and so on, is self-replicating, self-healing, and so on. And so in principle, you could take a small number of cells up with you, and you could learn to live off the land. Say you have photosynthetic cells, they can use the sunlight there. That's free. There's plenty of CO2 in the atmosphere. It's about 95% CO2, and then there's nitrogen in there, so you can fix the nitrogen as well. There's water, of course, on the planet. It's just not in a liquid form, so you need to melt it or you need to get it from the atmosphere, but you're better off probably melting it. And there are going to be minerals and so on in the regolith. So the materials are there for a cyanobacterium or an alga or something to be able to convert some of these raw materials into something that's usable. So that's really the crux of the whole thing, is that biology allows you to beat this up mass problem. Also, we have issues with storage. As I jokingly said, you can't just call Amazon.com and expect delivery the next day. Turns out this joke doesn't work so well in the UK because they don't get next day delivery either. But you know what I mean. You need to be able to store things. You need to be able to have flexibility. Again, you can't say, oops, I forgot my pipette min. You better have it with you or be able to use what you do have, and it better be reliable. So these are completely different challenges than my colleagues in other synthetic biology labs have when they need to look towards Earth things. So what's the big idea? Well, for millennia, we've used biology to do chemistry on planet Earth. There is absolutely no reason we shouldn't continue to use biology to do chemistry beyond Earth. We may use synthetically altered organisms for material production, including habitat construction and so on. I'll give you some examples in a moment. We may use material that's brought from the Earth to get started, but we may also be repurposing spacecraft, use materials that we find at destination, as the example that I gave you on Mars was alluding to, or maybe we pick up materials from an asteroid en route. We're going to use nanotechnology, and this is kind of an interesting one. I get invited to talk at interesting conferences, including the opening speaker for nanotechnology conference a couple of years ago. And I must say, the night before in my hotel room, I was thinking, what am I doing here? I couldn't even pass a freshman class in nanotechnology. And then I suddenly realized every cell in your body is a better nanotechnologist than any person who was in that room. Because we have to do nanotechnology at an incredibly precise level or we're not alive. And so if you use biology to do the nanotechnology, think about making DNA, RNA, making proteins. This requires incredible precision or you're no longer alive. So we can use biology to do nanotechnology. We may have alternate biochemistries. Everyone in this room uses right-handed sugars and left-handed amino acids. Why not make a mirror organism so we know what our worker organisms are, who's us and who's them, for example? We may send coding information digitally to Mars to make drugs or whatever. And this is a really important point, that we at this point have broken the physical link. So we no longer have to worry about physically having every sequence with us. We can come up with a new sequence, as our students do every day in the lab, on our computer, push send. That digital information goes to a DNA synthesis company, and then they send you the physical DNA. So now when you're talking about sending biological information digitally, you've bridged that gap between Earth and your sequencing or synthesis company, but also between Earth, the Moon, Mars, and so on. So that's huge that we have broken that physical continuity issue. We could actually potentially also generate electricity from organisms. Everyone who's still comatose in this room is generating electricity from chemical energy. So here is an example picture I took outside of Edinburgh, and you see lots of fantastic biological products that we use all the time, whether it's wood or wool or meat. Have a nice lamb shank or whatever. Now, we're not going to take any of these things with us. They're way too ridiculous to try to take, and it's too much upmass. We're going to take something like this. These are yeast cells. This is just baker's yeast that you could get to make bread or whatever. But you could then engineer them to make products like silk or wool or cellulose or latex, which a student of mine did last year, calcite, silica precipitation, spider silk, which is a very hot area right now because it's extremely strong. So there's several companies from the US and Japan, ones that I know about for a fact, that are making spider silk products. I mean, unbelievable stuff. You just can't domesticate a spider, it turns out. But this is, again, a great product, so you put it in a different form factor. So you take something like a yeast cell, and you now can produce these things off planet. And I have to say that I come from an evolutionary biology background. As many of you know, I was actually an undergraduate at Yale when there was a single biology department, but I would have been evolution and ecology. And so I grew up looking outside at nature and thinking, wow, you know, look at this amazing diversity. How did it get here? Where are we going? And so on. But then you switch hats as a synthetic biologist to a more engineering approach, and you look outside and say, wow, here's this amazing giant genetic hardware store. I can just start pulling things off the shelves and doing new things with it. And that's what we're talking about here. So, for example, habitats. Again, the Apollo astronauts lived in a tin can for a week, and you can just about do it when the whole world's watching and it's 1 week, and then you're going to be home and get a ticker tape parade. It's another issue if you're going to have a long-term settlement. And even with the current propulsion systems, we're talking about a year and a half on the surface of Mars, about 6 months to get there, a year and a half before the planet's realigned for your 6-month return journey. And a year and a half is a long time to live in a tin can. So the traditional ways to make habitats, to bring your spacecraft or whatever, our approach is to take organisms to agglutinate regolith and make bricks. And once you have a brick, like with this building, you can build an administration building, you can build a lab, you can do anything. And so this particular brick is not one we made, but it's made biologically agglutinating soils. Soil actually has a special definition with organics, so that's why I keep referring to Martian regolith, which does not have the organics. Another approach that we've started exploring and discussed with a few people last night is the idea of using microtexture. And so here's a drawing that was produced by an architect colleague who has a little firm, Red House, in Cleveland, and he's gotten into the microtexture biz, and we put together a proposal to make something like this habitat picture that you see. And the idea is you would have fungal spores between a double layer of plastic, and then this thing would inflate on Mars, and you would use cyanobacteria to, again, convert the raw material that you would get on Mars into something that the fungi can eat. Therefore, they produce these mycelia, these thread-like things that are basically like fungal roots, which can then be used to harden this shell. So they basically would be growing their habitat in place using the raw materials. This is the sort of thing that people have already started to do, and here are my colleagues in Cleveland, and you can see these long form factors that they've now made fungal mycelial boards with. There are a couple of companies like Ecovative in upstate New York that sells commercial products made with fungal mycelia, chairs. Actually, one of their great products is a packing material for wine bottles. Think about it, it's sort of like a completely biological, eco-friendly styrofoam. These are not synthetically altered, these are just as is. Biomining, we have a fabulous project going on in that. The traditional way to access metals is either to bring them or to use very heavy equipment. And again, you've got the upmass problem, so you don't want the bulky, heavy, expensive equipment. But biology is very good, think back to the nanotechnology comments, about very specifically binding metals. And if you're interested in the metals that we normally bind, look at the back of a vitamin bottle. Over 50% of the proteins in our body use a metal. And so if you can figure out how to access the ability to bind a very specific metal and then do something like engineer a bacterium with that motif, that short bit of the protein to very specifically bind the metals, you could, in principle, mine anything basically with chicken soup at room temperature. It becomes a totally different operation. That little cartoon shows you what we've been doing is adding this little metal binding protein to the flagella of a bacterium so they could swim through and bind specific metals. We've done copper, zinc, and nickel as proof of concepts. Now what's kind of cool is that we're particularly interested in applying that to urban biomining, which is simply taking metals out of spent electronics. Everyone in the room probably is sitting there with a pile of electronics. So what if when it's dead, instead of going off to some place places like India and China and Africa, where you can find very easily, if you Google online, these horrific pictures of people with pans of dead cell phones and so on, trying to get the metals out with the toxin generation just being really, really horrific. What if instead we could take the spent electronics, put them literally in a blender? What you have to do is dissolve the silica, which is fairly easy. There are organisms that make silicases, including sponges, and we have now transgenic silicase in the lab. And so you're dissolving away the silica matrix, and then you send waves of bacteria in to pull out the metals. And what's kind of cool is we've buddied up with a group on the fourth floor in our building who's making a plasma printer. So they then take the cells that are enriched, for example, in copper as the ink to actually go into this printer to print a new circuit. And what happens is during the printing process, the cells are dispersed, and so you literally just get the copper because the organic matter is degraded because of the plasma. So we then end up with this end-to-end recycling system for an integrated circuit, and there are two of the people working on it in my lab right now, eyeing a dead computer. No one's computers or cell phones will be safe in the lab at some point in the next few years, so we're going to have everything hidden, extra locks to keep them out. We have done some work with bioplastics, as many other people have as well. Microbial cellulose is something you don't even have to genetically engineer organisms to do. There are a couple of microbes that make absolutely pure cellulose. Cotton's about 95% cellulose, and this nanocellulose actually has other properties that make it particularly good for a lot of things, including things like wound dressings and so on. We used some of that for projects in our lab, and we sent some of this processed one on the right to the University of Edinburgh for their IGEM team in 2015, which they then tested to use for a biosensor to look for contaminants in heroin. So since we're no longer limited by the form factor to make these products, you can now add something like bioprinting. And one of our dreams has been in our lab to do something like make a sheet of a feather. Think about the vein from a feather. It's lightweight and flexible and strong. So what if instead of just plucking chicken feathers, you could literally make sheets of this material? Or another thing that we've been kicking around a lot is the idea of printing bone. Now, I realize that the Neanderthals did make bone habitats. My guess growing up in Connecticut is that you would have a lot of trouble getting a building permit for a bone habitat in Connecticut today. That's just a fictional example. In upstate Connecticut is not literally a bone house in upstate Connecticut. But it is a fabulous material. So imagine you could take the ability to do this, you print cells, and you make a sheet of bone that could then be used in architecture. And so maybe it's really going to be Back to the Future, where you can use the ability to make bone using something like a yeast cell to actually print materials there. Now, those are all examples of editing nature to make a specific product or to recycle. And I find that most people do not have issues with this. I have gotten some feedback, well, what if the organisms are loose? And if you're dealing with a biological product like a bone or spider silk or whatever, you're no longer talking about the living organisms once you're deploying it. And as I somewhat sarcastically tell NASA headquarters, it's sort of like leaving your sweater in the car overnight. I don't expect to find 3 sweaters there in the morning. They're dead. They're sheep products. They are no longer living sheep. But now I'm going to get into an example or two where we really are editing nature. Now, we're very interested in life in extreme environments, and that's partly from my astrobiology background, looking for life in the universe. But it's also incredibly important if we're going to take life off planet, and actually for a lot of industrial processes on the Earth. You may want to conduct them at a high temperature, high pH, low pH, or whatever. And so part of the 2012 team asked the question, could you make a synthetic extremophile? And this is a project they called the Hell Cell Project, and the name is stuck, and I absolutely love the name. And it's something that we're still very interested and active in. And this is the group of students from that year. You can see they're very energetic, full of beans and so on. So the Hell Cell toolbox that they came up with, and this is 4 students out of 12 in a 3-month project, were a few genes that should confer desiccation resistance, genes or operons, radiation resistance, pH, temperature, and cold resistance. And being of a certain age, I looked at this and I thought, this has got to be 5 PhDs, and there was no way, but they did every one of them, and every one of them helped push the limits for E. coli. Some of this was taken up with the 2014 team, where they were looking actually at biologically making a platform for exploration. In this case, it was a biodegradable drone. We did the balloons last year with embedded sensors. And the reason I wanted to show you this drone in particular is we started with the body of this fungal mycelium, covered it with the cellulose that you've now heard about, waterproofed it, and this is very cool, with taking a protein from wasp spit. And you think about it, wasp is the one organism in nature that chews up wood and spits out waterproof paper, and that's now turned into a senior honors thesis at Brown. But this is the one that then ties in with Hell Cell, is you want to then have a biosensor, but you don't want to, if it's used on the Earth, have something that if it crashes is going to interact with the organisms here. So what the students did is took the amberless Hell Cell, well, the amberless mutant that was created, I believe in George Church's lab, where it was a recoded cell, and then gave it some capabilities. And so the idea is that if this crashed and any of these cells got out, it was basically now on a different operating system than the rest of nature, and so there shouldn't be an interaction. We did not actually fly this or crash this, but this was in principle exploring the idea, and there was their little prototype that they made. So we've looked a little bit at this question of why you might want to do synthetic biology on Mars, and the economic also ties in with the fact that you're doing an end run, again, around the earthly economic systems. So let me finish by just sort of comparing the two issues. One is that we're worried a lot about human health and ecosystems, and again, that's what most other people are talking about in this conference. We actually have a little bit of that problem on Mars. The moon is considered uninhabited and uninhabitable and always has been in terms of an indigenous microbiome, but Mars is still an open question, and so there are planetary protection protocols. So instead of going the EPA and the FDA, there are international COSPAR treaties, Committee on Space Research, and so that is actually a concern with some bodies in our solar system and beyond, and we do have a planetary protection officer at NASA headquarters. The economics, as I said, is more of an upmass issue for Mars, for us, not the existing industries, but there are things like making wires out of DNA, which one of the IGEMers in 2013 did, which obviously has applications both on Earth and Mars, as many of these things do. Making biosensors, either molecular, whole cell, or even multicellular, can be done on the Earth or off Earth. And you get colored readouts, you get moving ones. I did have to throw in that one little picture of our self-folding origami. The picture on the top is a local company in Silicon Valley that's making tiny printable biosensors for the hands. And then we're applying this not just to Mars, but the USGS has us making some of these for looking at actually avian flu. Exploration platforms work perfectly well on the Earth as well as off planet. So a lot of this is interplanetary. But then finally, the philosophical questions, and again, other people have talked a lot about whether we should tamper with life on the Earth. Our big philosophical question is not tampering with life on the way. For example, I talked to one of the managers about GMOs and problems with people eating them, and he just laughed and he said, astronauts eat what they're told to eat. That's not a problem. But our philosophical problems are, what if there is an indigenous life form on Mars, or was one? As my astrobiologist speaking, if it turns out there was a second life form on the next planet over and we destroyed it, it would be one of the greatest tragedies, I think, in science ever, ever, ever. So we cannot do that. So my final thought was going to be, the can we, should we, and which most people have brought up, and I think that it's still an excellent point. So I decided to add some of these other questions, which I'm not going to discuss, but I'm hoping people bring up during the various discussions the next day and a half. And that is, what is natural? What is the difference really between domestication? There's nothing that we eat that we haven't already tampered with the genome. We just call it domestication. Is that really different in principle or scientifically from, for example, synthetic biology? And this you saw before. This is basically your cliff notes to the human ethics, so it answers all the questions that everyone else has posed. Thanks.